Hello everyone, welcome into the ITLS Academy. Myself, Dr. Himani Singh, and here's my mobile number and mail ID is given. And if you have any query in this lecture, then you can call me on this number and you can mail me on this mail ID. So today we are going to just discuss about the stem cells. Actually, what is the stem cells? We need to make new cells all the time just to keep our body functioning. Some specialized cells such as blood and muscle cells are unable to make copies of themselves through cell division. Instead, they are replaced from population of stem cells. So, we can say like that stem cells having unique ability to produce both copies of themselves and other more specialized cell types discard differentiations every time they divided. And stem cells therefore are essential to the maintenance of tissues such as blood, skin and gut that undergo continuation turnover, cell replacement and muscles which can be built up according to the body's need and is often damaged during physical exertion. Now question is this, what is the exactly definition of stem cells? So we can define as a stem cells like stem cells are undifferentiated mass of cell that has the ability to continuously divide and differentiate, develop into various other kind of cells and tissue. The human body has a variety of 220 different cells types. Stem cells are master cells that act as foundation cells for every organ, tissue and cell in the body. They are programmed to perform particular tasks and they serve as a repair machine for the body. Here we can see the diagram like stem cells is the self-renewal cells and differentiate into the different type of mature cells. Now here we can see this history about the stem cells. In 1998, researchers first extract stem cells from human embryos. In 1999, first successful human transplant of insulin making cell from cadavers, and 2001 President Bush restricts federal funding for embryonic stem cells research, and 2002 Diabetes Research Foundation International created $20 million funds raising a force to support stem cells research. 2002 California stem cells research, and 2004 Harvard researchers grow stem cells from embryos using private funding, and in the 2004 ballot measures for $3 million found for bond for stem cells. Now question is that what makes stem cells unique? In this picture you can see here the stem cells could be the self-renewal and is differentiate into the specialized cells and self-renewal and again convert into the stem cells. So first we will discuss about the stem cells are the unspecialized cells. Unlike a red blood cells which carries oxygen through the bloodstream or a muscle cell that work with other cell to produce movement, a stem cells does not have any specialized physiological property. Second one is the self-renewal is the defining property of stem cells. So, stem cells can divide and produce identical copies of themselves over and over again. Specialized cells such as blood and muscles do not normally replicate themselves, which means that when they are seriously damaged by disease or injury, they cannot replace themselves. Third one, we will discuss about the stem cells can also divide and produce more specialized cell types. This procedure is called differentiation. So, stem cells from different tissue and from different stages of development vary in the numbers and types of cells that they can produce. Features of the stem cells. First features is stem cells are very unique cells. Second features is stem cells have the amazing ability to develop into several distinct cell types in the body. And third one is the stem cells can be used as a repair system for the body. Fourth one is the stem cells can theoretically divide without limit in a living organism in order to replace various type of cells. And fifth one is the when a stem cells divide, each new cells has the potential to either remain a stem cells or become another type of cells with a more specialized functions like muscle cells or red blood cells or brain cells, etc. Now here we can see in this picture different stages of development of stem cells. Now first stage is the fertilization, and then second stage called zygote. After fertilization, second stage convert into the zygote. This called as totipotent cells. We will discuss in the further slides. And third stage is the morula, and fourth one is the blastocyte, and fifth one is the epiblast cells, and this one fifth one is the pluripotent stem cells. This condition is called pluripotent stem cell. This one you can see by arrow, this is the epiblast, and this layer is called the hypoblast, and then it converts into the sixth stage is the fetus. So this natural stem cells just made by the morula, blastocytes, and this epiblast and hypoblast cells and fetus cell. Then by these procedure we can isolate of the stem cells for the artificial stem cells synthesis. In this picture also we can see different stages of the development of stem cells very clearly. In this first stage you can see here in the single cell embryo, after 3 days this single cell embryo convert into the again look like an embryo, but this pictures look different types, and in this into the 5 to 7 days this is the picture, and 4 weeks this picture convert into this stage, and 6 week almost embryo can create it. So 5 to 7 days embryo called is the embryonic stem cells. This embryonic stem cells called the totipotent stem cells. We will discuss in the further slide what is the totipotent stem cells and pluripotent stem cells and multipotent stem cells. So just we will discuss briefly in the next slides. And in the 6th week, this conditions called embryonic germ cells. This conditions called is the pluripotent cells, and this fetus after 6 weeks, this fetus tissues stem cells could be the pluripotent or the multipotent. Here you can see this infant convert into the adult. So the stage of infant and adult, in the middle stage of the infant and adult, this called the adult stem cells. It could also be pluripotent and the multipotent. And here you can see this adult stem cells called is the embryonic carcinoma cells. These cells are the pluripotent stem cells. Now we will see by this picture different types of stem cells. So stem cells differentiate into two types. First one is the based on differentiation potential, and second one is the based on origin. So based on differentiation potential, first one is the totipotent, next one is the pluripotent, and third one is the multipotent, fourth one is the oligopotent, and fifth one is the unipotent. By the based on origin, you can see here it's divided into different types. First one is the embryo, next one is the fetus, perinatal is called infant, adult cells, and iPS cells. This is called the so in this picture we can see here again fetus divided into the three cells. This is a neural creates feature hematopoietics and again fetal mesenchymal stem cells, and infant convert into again divided into again umbilical cord, amniotic fluid, and placenta. Adult stem cells again divided into the hematopoietic stem cells, mesenchymal stem cells, neural stem cells, epithelial stem cells, hepatic stem cells, and pancreatic stem cells. Now we will discuss or classify with the first thing based on differential potential. So let's classification of the stem cells. First one is the what is pluripotent. Pluripotent stem cells able to differentiate into almost all cell types. Example is cell divides from mesoderm, endoderm, and ectoderm. Totipotent cells able to differentiate into any other types, like zygote form at egg fertilization. Third one is the multipotent cells, able to differentiate into closely related family of cells, like hematopoietic stem cells. Example is like the hematopoietic stem cells. And now, what is the oligopotent cells? This oligopotent cells able to differentiate into a few different cell types, like lymphoid and myeloid stem cells. And next one is the unipotent, able to produce only their own type of cells, like muscle stem cells. So here a small description of the stem cells based on differential potential. So now we will discuss briefly about this differential potential stem cells. First one, we will go to discuss about the totipotent stem cells. So with regards to stem cells, potency refer to their ability to differentiate into distinctive types of cells, whereas some stem cells are capable to generating more type of specialized cells. Specialized cells means those with greater potency. Other can only generate few specialized cells and thus have lower potency. So, classification of stem cells on the basis of potency. Stem cells can be classified by the extent to which they can differentiate into different cell types. Here is the five type of stem cells. First is the totipotent, next one is the pluripotent, multipotent, unipotent, and next one is the oligopotent. So first we will discuss about the totipotent stem cells. So totipotent stem cells is a good example of a totipotent stem cells. These cells are characterized by high potency, which means that they are capable of differentiating into any type of cell in an organism. For instance, the zygote formed following fertilization of the egg give rise to type of cells that form particular tissue and organ. The word toti means is whole. So this means totipotence is means whole cell. So totipotent stem cells are located in the early cells of a fertilized egg. Now we will discuss about the pluripotent. Example of pluripotent stem cells include embryonic stem cells. Compared to totipotent stem cells, pluripotent stem cells can only progress to cell from any of three germ layer. First is the mesoderm germ layer, second one is the endoderm germ layer, and third one is the ectoderm layer. Unlike the totipotent stem cells, pluripotent stem cells are located in the inner mass of the blastocyte. Pluri means many. So here we can differentiate. Toti means a whole, but pluri means many. So this is the difference in totipotent and pluripotent stem cells. Now we will discuss about the multipotent stem cells. Multipotent stem cells are the type of stem cells that only produce closely related cells. That means hematopoietic stem cells, for instance, can only produce red and white blood cells, including platelets. As such, they do not differentiate into a wide variety of cells, as is the case with totipotent and pluripotent stem cells. Now, here, as we can see here, hematopoietic stem cells, for instance, can only produce red and white blood cells, in which including platelets. So because these types of stem cells yield to very specific type of cells closely related, they are found in many tissues in the body where they perform this function. Multi means several. So now we can differentiate three cells. First one is the totipotent. The toti means is wholes, pluri means many, and now multi means several. Now we will discuss about the oligostem cells. Stem cells like myeloid stem cells are referred to as oligopotent because they produce a few types of cells like neutrophils and eosinophils. Now we will discuss about the unipotent. Unipotent stem cells give rise to cells of their own type along a single lineage. As such, unipotent stem cells have the lowest differentiation potential compared to other type of stem cells. Skin and muscle cells are a good example of a unipotent stem cells. Now we will discuss about the classification based on the source. So based on the source, the stem cells are widely divided into three main categories. First one is the embryonic stem cells. So as the name suggests, embryonic stem cells are the type of stem cells derived from embryos. It's like 4 to 5 days old. We have already seen in this previous picture. During days 3 to 5 following fertilization and prior to implantation, the embryo at this stage called a blastocyte. We can see in this picture, this one, in this picture we can see this 6 is the 6 week is the embryo, and this is the blastocyte. So you can see during day 3 to 5 following fertilization, in the day 3 to 4 divide the fertilization, and prior to implantation, the embryo at this stage called is the blastocyte. And this blastocyte contained inner layer cell mass that is capable of generating all the specialized tissue that make up the human body. Again, I am repeating, this blastocyte inner layer, are capable of generating all the specialized tissue that make up the human body. Embryonic stem cells have the ability for self-renewal, and the fact that they differentiate into every type of cells in the body. These stem cells significantly contribute to the growth and development of a fetus in a womb. These are purely present during the first stage of these stem cells significantly contribute the growth and development of a fetus in the womb. These are only present during the first stage of the development, and the embryonic stem cells are derived from the inner cell mass of embryo that has fertilized in vitro and donate for research purpose. Following informed consent, embryonic stem cells are not derived from eggs fertilized in a womb body. At about the third day of division following fertilization, in this picture you can see here, at about the third day of division following the fertilization, at the 8 cell stage, this 8 cell stage, the cells are totipotent, and this stage the embryo is capable of developing into a full developed organism. In this stage, the embryo is capable of developing into a fully developed organism. The blastocyte is formed about the 5th day. In this picture, you can see this the blastocyte is formed about the 5th day at this point. At this point, the inner cell mass that contains the embryonic stem cells, pluripotent stem cells, is fully formed. In this picture, you can see here, this is the egg, and this again egg convert into the zygote, and zygote divided into 2 cells stage, and again this divided into the 4 cell stage, and this 4 cell stage convert into the morula, and then blastocyte formation is started. This is the inner side of the blastocyte. And now in this picture you can see here, in the day first zygote is here, then again zygote start to dividing into 2 part. Again this is the day 1 or day 2, this is the day 2 and 3. 4 cells are divided into 2 and 3 in the 2nd or 3rd days, and in the days 4 or 5th morula is formed here, and again next day 5th or 6th blastocyte is formed here, and this is the inner structure of the blastocyte. Here you can see this picture of the embryonic development. This is the fertilized egg, this is the 2 cells, then 4 cells divided, and again at this cells fertilized egg divided into the 8 cells, this called blastocyte. This again divided into 16 cells stage, and this blastocyte is here. And then the this is the fetus 4 week, this is the fetus of 10th week, and this is the growth of 16-week old fetus. And in 20 days week, in the 20th week, this is the complete fetus here. Now we will discuss about the second type of stem cells, is adult stem cells. So adult stem cells are undifferentiated cells found living within specific differentiated tissue in our bodies that can renew themselves or generate new cells that can replace dead or damaged tissue. You may also see the term somatic cells used to refer to adult cells. The term somatic refer to non-reproductive cells. Again I am repeating, the term somatic refer to non-reproductive cells in the body. This means eggs or sperms. It could be the eggs or sperms. Unlike embryonic stem cells that are derived from a mass of cell of the blastocyte, adult stem cells can be found in different part of the body. Adult stem cells are typically scarce in native tissue, which have rendered them difficult to study and extract for research purpose. This makes it possible to replenish and regenerate tissue in case where cell die or damage. So, reside in most tissue of the human body, discrete population of adult stem cell generate cell to replace those that are lost through normal repair, disease or injury. So, adult stem cells are found throughout one lifetime in tissue such as the umbilical cord, placenta, bone marrow, muscles, brain, fat tissue, skin, gut, etc. The first adult stem cells were extracted and used for blood production in 1948, and this proceed was expanded in 1968 when the first adult bone marrow cells were used in clinical therapies for blood disease. Now we will discuss about the types of adult stem cells. Here the 5 types of adult stem cells are present here. First one is the hematopoietic stem cells, second one is the mesenchymal, third one is the neural, and fourth one is the skin, and next and last one is the induced potent stem cells. Types of adult stem cells are only 4 types. First one is the hematopoietic, second one is the mesenchymal stem cell, third one is the neural stem cells, and fourth one is the skin stem cell. Induced potent stem cells are the type of only stem cells. We have already discussed in the previous slide here. So classification based on the source. So here the classification based on the source. First cells are embryonic stem cells. Second one cells are adult stem cells. We have already discussed about those 2 types of cells, and third one is the induced pluripotent stem cells. We will discuss into the next coming slides. So first we will discuss about these 4 types of the adult stem cells. Now we will discuss about the hematopoietic stem cells. Hematopoietic stem cells formation start during embryonic development. Hematopoietic stem cells are the cells that give rise to myeloid and lymphoid cells from the bone marrow and peripheral tissue respectively. It can become a common myeloid erythroid progenitor which give rise to all blood cells like granulocyte, monocyte, and macrophages, or it can become a common lymphoid progenitor, which is called CLP, which give rise to B lymphocyte and natural killer cells. You can see in this picture, blood stem cells is here, is divided into the myeloid stem cells and the lymphoid stem cells. Myeloid stem cells again divided into the red blood cells, platelets, and it convert again into the myeloblast. Myeloblast form the white blood cells, and here the lymphoid stem cells give the formation of the lymphoblast, and this lymphoblast again convert into the white blood cells. So we can define hematopoietic stem cells as an immature cell that can develop into all type of blood cells, including white blood cells, red blood cells, and platelets. Hematopoietic stem cells are found in the bone marrow, umbilical cord blood, and peripheral blood, also called blood stem cells. Hematopoietic stem cells only make up a small percentage of cell in the bone marrow. This about 1 in 10,000 cells. So, in addition to the ability to renew themselves, this is called self-renewal. And because these help to produce only specific related cells, blood cells, they are described as multipotent stem cells. Now we will discuss about the mesenchymal stem cells. Mesenchymal stem cells are an example of tissue or adult stem cells. Mesenchymal stem cells in the adult bone marrow are necessary for the body to generate tissue such as bone, cartilage, muscles, ligament and tendon, adipose and bone marrow. They are multipotent, meaning they can produce more than one type of specialized cell of the body, but not all type. You can see in this picture, this is the bone marrow. This convert into the mesenchymal stem cells. This mesenchymal stem cells is the self-renewal cells. This differentiate into the adipose, chondrocytes and osteocytes. Osteocytes is present in the bones, chondrocyte present into the cartilage, and adipose present into the fat. So in this picture we can see the mesenchymal stem cells can make fat, cartilage and bone marrow. They have not been proved to make other type of cells and of the body. So mesenchymal stem cells make the different specialized cells found in the skeletal tissue. For example, they can differentiate or specialize into cartilage cells. We have already seen in this picture. So, cartilage cells is the chondrocyte cells, bone marrow cells is the osteoblast cells, and the fat cell is the adipocyte cell. So, these specialized cells each have their own characteristic shape, structure and functions, and each belongs in a particular tissues. Some early research suggests that mesenchymal stem cells might also differentiate into many different types of cells that do not belong to the skeletal tissue, such as nerve cells, heart cells, liver cells, and endothelial cells, which form the inner layer of the blood vessels. Now we will discuss about the neural stem cells. So, neural stem cells are self-renewal. multipotent cells. Neural stem cells are the stem cells of the nervous system. During development, they give rise to the entire nervous system. These stem cells are found into two zones of the dendritic part of the hippocampus in the brain, as well as in the striatum part of the basal ganglion located deep within the cerebral hemisphere. The neurocortex, the outer layer thickness of the highly convoluted cerebral cortex, and the spinal cord. Here in this picture you can see here neural stem cells divided into the neuronal processor, and here is the glial processor. So neural processor convert again divided into the sensory neurons and motor neurons, and here you can see in the is converted into the interneurons and multipolar neurons. So precursors again divided into the astroglia and oligodendroglia. So now you can able to understand about the neural stem cells. Now we will discuss about the epithelial stem cells. As compared to various multipotent stem cells, epithelial stem cells have been shown to regional specific. Through stem cells marker, epithelial stem cells have been identified in such regions as the basal layer, sebaceous gland, and kidney papilla, among others. Epithelial stem cells give rise to epithelial cells, which constitute 60% of the differentiated cells in the body, responsible for covering the internal, like interstitial lining, and external surface, like skin, of the body, including the lining of vessels, glands, and other cavities. So, like other stem cells, epithelial stem cells are also capable of self-renewal while continuing developing into various mature cells that serve different functions. In the gut, epithelial stem cells produce several types of cells, including goblet cells, patent cells, and absorptive cells, among others. Epithelial stem cells are involved in the maintenance homeostatics as well as the normal functioning of various tissue and organs such as the lung. Now we will discuss about the induced pluripotent stem cells, third type of the stem cells. So, including pluripotent stem cells, immature cells that is generated from the adult, like mature cell, and that has regained the capacity of differentiate into any type of cell in the body. So as compared to the other type of stem cell in the body, induced pluripotent stem cells are unique in that they are made in the laboratory. Here embryonic genes are introduced into the somatic cells, which convert them into stem-like cells. Therefore, induced pluripotent stem cells are differentiated from a majority of stem cells that are produced in various parts of the body. However, like the other, these are capable of giving rise to various specialized cells. Now in this picture you can see here starting cells from the donor tissue. This is the donor person. So include change in the gene expression at this induced pluripotent stem cells, structure of the pluripotent stem cell. So stem cells, induced pluripotent stem cells derived from the adult cells in 2007. It was very recent discovery. So can be grow indefinite in culture in an undifferentiated stage. And similar properties to the embryonic stem cells as can differentiate into many different tissue type pluripotent, and induced pluripotent stem cells can create stem cells directly from a patient for research. Now we will discuss about the skin stem cells. So this is the part of, you can say, skin stem cells is the type of adult stem cells. So the epidermis of the skins and the hair follicles provide a protective barrier against microbes, injury, and discretions of underlying cells. Skin cells are continuously sloughed so integrally of this barrier is entirely dependent on regular cell renewal. So the question is, what do we know about the skin stem cells? First is, skin is a special organ that both protect us and allow us to sense the world around us. Skin is made of three layers, each containing specialized cells. A variety of stem cells are needed to maintain and repair our skin on a daily basis. Researchers have identified stem cells responsible for making the epidermal layer, hair follicles, and skin pigments. Epidermal stem cells are currently used in clinics to grow skin for patients with life-threatening burns. However, the process is difficult, expensive, and the skin is not normal. It lacks sweat gland, hair follicles, and sebaceous gland. So the homeostatic function is carried out by skin stem cells that reside in the epidermis, the hair follicles, and sebaceous gland, which proliferate in response to external signals. Researchers are currently working to develop methods to grow skin that contain more of the normal functional components such as sebaceous gland, hair follicles. This will make skin graft more durable and natural looking. Now we will discuss about what are the potential applications for stem cells. First, we will discuss about the stem cells can be used to study development. Stem cells may help us understand how a complex organism develops from a fertilized egg. In the laboratory, scientists can follow stem cells as they divide and become increasingly specialized, making skin, bone, brain, and other cell types, identifying the signals and mechanisms that determine whether a stem cells choose to carry on, uh, replicating itself or differentiate into specialized cell types, and into which cells type will help us understand what control normal development. Some of most serious medical conditions such as cancer and birth defect are due to abnormal cell division and differentiation. A better understanding of the genetic and molecular controls of these processes may yield information about how such disease arise and suggest new strategy for therapy. This is an important goal of the stem cells research. Second application is stem cells have the ability to replace damaged cell and treat disease. So this property is already used in the treatment of extensive burn and to restore the blood system in patients with leukemia and other blood disorder. Stem cells may also hold the key to replacing cell lost in many other devastating diseases for which there are currently no sustainable cures. Third one is the stem cells could be used to study disease. In many cases, it is difficult to obtain the cells that are damaged in a disease and to study them in details. Stem cells either carrying the disease gene or engineered to contain disease gene offer a viable alternative. Scientists could use stem cells to model disease process in the laboratory and better understand what goes wrong. Stem cells could provide a resource for testing new medical treatment. Now, in the last, we will discuss about the stem cell therapy. So, stem cells may be one way to generate a new cell that can then be transplanted into the body to replace those that are damaged or lost. Adult stem cells are currently used to treat some conditions, for example, like blood cells. So, blood cells are used to provide a source of healthy blood cells for people with some blood conditions such as thalassemia and cancer patients who have lost their own blood stem cells during treatment. Second one is the skin stem cells. Skin stem cells can be used to generate new skin for people with several burns. In the age-related macular degeneration is an example of disease where stem cells could be used as a new form of treatment in the future. So, some people with age-related macular degenerative lost their sight because cell in the retina of the eye called retinal pigment epithelium cell stop working. Scientists are using induced pluripotent stem cells to produce pigment epithelial, retinal pigment epithelial cells in the lab that can then be put into a patient eye to replace the damaged cell. Next, use of stem cell therapy. Stem cells could be used to generate new organs for use in transplant. Currently, damaged organ can be replaced by obtaining healthy organs from a donor. However, donated organs may be rejected by the body as immune system sees it as something that is foreign. So, include induced pluripotent stem cells generated from the patient themselves could be used to grow new organ that would have a lower risk of being rejected. So guys, I have already covered almost things related to stem cells. So this is the basic lecture about the stem cells. If you have any query, then you can mail me or you can call me, and you can just follow us our academy by the this Instagram, Facebook, Twitter, YouTube, and WhatsApp, and by the LinkedIn. Thank you. I hope you will enjoy this lecture. So, guys, we will meet you in the next lectures. Thank you again.